Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to our Pathology. In previous video, we were started apoptosis. We were discussed some causes of apoptosis, that in which conditions apoptosis occurs, what are the situations in which apoptosis occur, and we were also discussed some morphological changes of apoptosis. The link for that video is in description box. You can check it there. Now we proceed toward mechanism of apoptosis. In today's video, we will discuss about mechanisms of apoptosis. There are two general major pathways of mechanism of apoptosis: intrinsic pathway, and the other one is extrinsic pathway. Intrinsic pathway, why it is called intrinsic pathway? Because the origin of this pathway is inside cell. This pathway originates from mitochondria. That is why it is also called mitochondrial pathway. The other one is extrinsic pathway. The origin of extrinsic pathway is outside cell. It is originates from outside cell. That is why that is called extrinsic pathway. In today's video, we will only discuss about intrinsic pathway, and inshallah in the next video we will discuss extrinsic pathway. Before we proceed towards intrinsic pathway, let's first discuss about caspases. Caspases are proteins or enzymes inside cell that it is required for apoptosis. It is necessary to activate these proteins to undergo apoptosis. So for apoptosis to occur, it is necessary to activate caspases. Caspases are cysteine proteases. If we look at the word caspases, C is for cysteine, ASP is for aspartic acid, and ases. So these are cysteine proteases which cleaves cysteine after aspartic acid residues. So that is why it is called caspases. So for apoptosis, it is necessary to activate caspases. So the ultimate result of all pathways, either it is intrinsic or extrinsic, is the activation of caspases. Ultimately, there are activation of caspases. So after activation of caspases, apoptosis occurs. Now let's discuss about pro-apoptotic and anti-apoptotic proteins. Pro-apoptotic proteins are those proteins which activates or which stimulates apoptosis. When pro-apoptotic proteins are activated, it stimulates apoptosis. On the other hand, anti-apoptotic proteins, these are those proteins which inhibits apoptosis. When these proteins are in inactive form, these inhibits apoptosis. For both intrinsic and extrinsic pathway, there are some pro-apoptotic and anti-apoptotic proteins. For intrinsic pathway, there are a family of proteins called BCL2 family. BCL2 family contains both pro-apoptotic, anti-apoptotic, and some sensor proteins. For intrinsic pathway, pro-apoptotic proteins are for this pathway. Pro-apoptotic proteins are BAX and BAC, and anti-apoptotic proteins for this pathway is which inhibit this pathway are BCL2 itself and BCLXL. So these are pro-apoptotic proteins for this, and these are anti-apoptotic proteins for intrinsic pathway. Now let's start intrinsic pathway. First of all, when there is some injury occurs, some damage occurs, so cells have the ability to sense a damage. So there are some sensors inside cell which sense the damage to the cell. For example, if there are DNA damage occurs or some growth factor withdraw some growth factors, there is a signal of cell death. So the cell will sense that damage. There are some sensors called Bh3 proteins. Bh3 proteins is also related to BCL2 family. So these Bh3 proteins sense damage to the cell and it activates pro-apoptotic proteins. As I said already that Bh and Bh are pro-apoptotic proteins. So these apoptotic proteins are activated. When these sensors sense the damage to the cell, it activates Bh and Bh, which are related to BCL2 family. These are effectors of BCL2 family. And on the other hand, these sensors can also inhibit anti-apoptotic proteins called BCL2 and BCLXL, because there are a signal of cell death or a signal of apoptosis. So these anti-apoptotic proteins must be inhibited. So BCL2 family effectors are activated and it in turn activates or it form channels in mitochondria. This is mitochondria. So these Bhs and Bh insert in membrane of mitochondria and form channels. These are channels in mitochondria. And when channels form in mitochondria, some proteins leak out. Through these channels, some proteins leak out. For example, cytochrome C and as well as some pro-apoptotic proteins leak out through these channels. And these proteins will stimulate apoptosis. So when these proteins and cytochrome C leaks out, it activates initiator caspases, which converts to executioner caspases. Initiator caspases are actually these are in inactive form. These are those caspases which are in inactive form. So when these are activated by cytochrome C and pro-apoptotic proteins, it becomes executioner caspases, which executes apoptosis. That's why it is called executioner caspases. For example, if there are pro-caspase 9, this is an initiator caspase because this is not in inactive form. When it is stimulated or activated by pro-apoptotic proteins, it becomes caspase 9. It becomes caspase 9. Caspase 9 is an active form. It can do apoptosis or it can stimulate apoptosis. So caspase 9 is an executioner caspase. So these different caspases are activated, and now these executioner caspases will activate enzymes inside cell. Like it can activate endonuclease. Endonuclease is an enzyme which can cleave DNA. So this caspase will activate endonuclease. Endonuclease activates and it will like DNase. So this DNase will go inside cell and it will degrade DNA. So if DNA degrades, it will destroy the cell. On the other hand, it can activate protease activation. And as we know that protease is an enzyme which can cleave proteins, which can degrade proteins. And as we know that if this is a cell, this is a nucleus, inside cell there is a skeleton called cytoskeleton. This is cytoskeleton. This cytoskeleton gives a proper shape to a cell and also helps in movement of a cell, give proper structure to a cell. So if these caspases activate a protease, this protease will come and will degrade this cytoskeleton. So if this cytoskeleton degrades, cell structure disturbs, cell distorts, cell will become like this. So cell shrinks and apoptosis occurs, and ultimately cell will phagocytosed through macrophages. Now you must understood one point, that how these proteases are activated. So let me draw. For example, this is an protease. This is a protease. Normally it is blocked by some inhibitor. This inhibitor, it blocks this protease. When case phase are activated, case phase comes and remove this inhibitor from enzyme, and its active sites become empty. Now it can work. Now it is activated. Now it will come and will degrade proteins. So the cell will destroy. Cell will move forward towards apoptosis. So this is our today's lecture. If you people understood this, you can subscribe my channel and also press the bell icon. So thank you.